Lecture 6 will be the first in our series on the cellular metabolism, and it'll be divided into 3 sections: an introduction to metabolism, a review of ATP and energy coupling, and finally a deeper look into enzymes. First, start by answering these 5 review questions on Socrative. Pause the video and come back when you're done. When we look at both phospholipids and integrins, although both of these are components of the membrane, they're made of different macromolecules. Phospholipids are going to be lipid components, while integrins are proteins that are embedded into the membrane, spanning from the extracellular matrix into that inner cytoplasm. Both of these are going to need to be able to interact with both the hydrophilic regions on either side of the membrane and the hydrophobic core. And the term that we use to describe that is amphipathic. We're going to continue to be thinking about the fact that these membranes are composed of both lipids and proteins like integrins, which are capable of doing jobs. Another example of this job would be a transport protein, which can be a component of either passive or active transport. When we differentiate between active and passive transport, it's important to note that their difference is not based on the presence of a protein. As a matter of fact, there are forms of passive transport, like diffusion or osmosis, which do not require a protein to be present to help move components across the membrane. Passive and active transport are really differentiated by the direction in which the molecule is transported across the membrane. Active transport goes against the gradient, while passive transport goes down the concentration gradient. And so one of the other ways, an extension of this, is to recognize that active transport requires energy be put into the system, most often ATP, in order to move solutes against their gradient, where passive does not, because it's moving in the direction that they would naturally move. An electrochemical gradient is going to be a special kind of gradient, composed of both electrical components and differences in a concentration gradient. That's where the term electrochemical comes from, is a recognition that there are 2 components here. Which of the following describes a hypotonic solution? Well, hypo means less. So we're going to be dealing with a solution that has less solute. I do want to recognize, however, that although there is less solute, the most important is to recognize what that solute concentration does to free water. If we have less solute, we have fewer hydration shells forming around that solute. Fewer hydration shells mean that there's more free water. So what would happen to an animal cell that was in this environment? Well, you would have a swelling result because all that free water from outside of the cell would move into the cell via diffusion or osmosis, and then this would cause the cell to swell and eventually burst. A plant cell, which has a cell wall, would form what we know as turgid, preventing this lysis, but it would not change this osmotic action of a cell in the hypotonic solution. You would still have the rush of that excess free water from the solution into the cell. Today we're going to be identifying molecules that are high in free energy, justifying that selection, and then talking about that free energy's ability to do work inside the cell. We need to describe energy requirements for chemical reactions, describing them as spontaneous, exergonic, or endergonic to be able to understand the impact of energy on these reactions. You should be able to draw a chemical reaction scheme as well as describe how ATP facilitates chemical reactions, and do the same for enzymes, and compare and contrast the difference between these 2. And then you will describe factors that can influence and or regulate enzyme activity as an important component for how we regulate metabolism in a cell. So there'll be 3 videos, the first of which is going to be talking about an introduction to metabolism, and recognizing that a metabolism is the totality of an organism's chemical reactions. Some reactions will extract energy from food, while others will use the energy that's been extracted to do work inside of the cell. Here's a great opportunity to challenge yourself and try to connect what we're doing today with things that we've already done in this class. Pause the video here and create a list for yourself. What work does the cell need to do? I'm never going to go through this specifically, nor will your textbook, but you'll be asked questions, such as on your final exam, where I might ask you to connect metabolism to other organelles in the cell, for example, or different macromolecules. So challenge yourself to do that here, and you'll already be a step ahead. To better understand metabolism, scientists organize these different reactions into pathways. It allows us to more easily recognize how we can get from a starting molecule to a final end product. Now, each step in a metabolic pathway is going to go through and utilize a specific and unique enzyme. So in order for us to get from a starting material to an end product, we're going to have to go through a series of reactions, each utilizing a different enzyme. Catabolic pathways release energy by breaking down complex molecules. The same principles apply. You use an enzyme at each step as you systematically break the molecule down. But as with cellular respiration, we're going to be going from a large molecule of glucose, utilizing oxygen in the process, and releasing energy in the end as one of our products. It's an example of catabolism that we're going to be looking at in more detail in our next class. Anabolic pathways, on the other hand, consume energy, and they can build larger molecules from simpler ones. For example, the synthesis of polypeptides from amino acids is a catabolic pathway. This is an example of work that the cell has to be able to do. In order for us to better understand metabolism, we really have to talk about energy, because all of these reactions have to do with energy. And energy is the capacity to be able to cause change. So the type of change that we cause to occur will tell us the type of energy that's being used. An example of energy that we already know and have talked about with water would be thermal energy. It's a form of kinetic energy, which is associated with the movement of atoms or molecules. An example of this would be heat. And this is the reason why heat is capable of disrupting hydrogen bonding in a water solution. In this class, we're going to talk much more about potential energy and that can possess the ability to do things. Chemical energy is going to be the form we focus on most because it's potential energy which is available for release in a chemical reaction. And that's what metabolism really is. Now, when we talk about biology, it's important to remember that this is going to be governed by particular physics laws. Specifically, the first law of thermodynamics. It states that energy can be transferred or transformed, but never created or destroyed. So in none of the reactions we look at today is energy going to magically appear or disappear. So if you have a high free energy molecule, that energy is not going to disappear. It's going to have to be transferred somewhere. Now, biologists start by understanding how that transfer is going to occur. And we do this by deciding whether or not a reaction is spontaneous or non-spontaneous. Spontaneous means that reactions transfer energy without first requiring the addition of energy. You can think of this as something that will proceed on its own. That's why we use the word spontaneous. An example would be a rock at the top of a hill. A spontaneous reaction means that the rock will slide down that hill. You don't have to do anything. It likes to move downhill. This can be fast or slow. If you have a very shallow hill, it might take hundreds of years for that rock to slowly work its way down. If it's a very steep hill, it could be a matter of seconds. Spontaneous does not tell us anything about the rate of the reaction, only that the reaction will proceed forward and we don't have to add any energy to do so. This is determined by the amount of free energy that are in the reactants. Free energy is energy that can do work. So there's going to be some component of energy to the cell, but the amount that's free is going to be the amount that can do work. You can think of this a little bit like free water when we looked at osmosis. Some water was being taken up by hydration shells, and so it couldn't diffuse down the concentration gradient. Well, energy can also be committed to doing work, and so free energy is the energy that's available. Systems will always go towards the more stable, less energetic state. That less energetic state is going to have lower free energy. An example is diffusion. It's a lot of energy to pack all that free water close to each other, as where when you diffuse down your concentration gradient or to a more hypertonic solution with lower free water, now you're dealing with the ability for water to space itself out. That's lower free energy. When we're dealing with molecules, large complex molecules with lots of bonds and lots of charges are going to be higher in free energy. Smaller uncharged molecules are going to be more stable and lower in free energy. You might have already seen this principle before in chemistry. Spontaneous reactions are also known as exergonic. An exergonic reaction will proceed with a net release of free energy. And an endergonic reaction is non-spontaneous and will absorb energy from its surroundings. In general, it's most appropriate to use the term spontaneous or non-spontaneous when you're talking about the ability of a reaction to proceed on its own. But when you're specifically talking about energy, it's favorable to use the terms exergonic or endergonic because these specifically deal with this addition or release of energy from the system.